with magnetic tweezers. So just a very, you've heard about these from Pavel actually only, I guess, a sort of week or so ago. But anyway, these magnetic tweezers involve taking a magnetic particle, dynabead, which many of you use actually for purifying proteins and things. And these dynabeads are derivatized, and these particular ones we have with streptavidin on the outside. And they will bind to DNA, which has been modified to have biotin on one of its ends. And on the other end of the DNA strand, we put digoxygenin, and that will then bind to anti-digoxygenin, which is put it on the microscope cover slip. So now you come along with your magnet, and the bead pops, the magnet's above here, and the bead pops up, stretches out the piece of DNA, and now we're actually being able to manipulate a single strand of DNA. And so the beauty of this method is that you're doing a single molecule experiment, but you actually use rather crude techniques to prepare the sample. You just take the beads, mix them with DNA, and you chuck them into your microscope slide. You wash out all the junk, and you're left with these lovely little things here. I say it's easy. In fact, Algae spent about a year getting rid of non-specific binding, multiple tethers and all this sort of stuff. So there is quite a lot of startup to getting it working. But anyway, then you can land up with a piece of DNA, a piece of plasmid DNA, with your origin site, this thing called ORI-D that Rep-D binds to, stick that in the middle, and now we can try looking at this initiation process. So how are we doing that? Well, we're using, so this is just demonstrating the stretching business. So as we come down with our magnets, the beads pop up, and you can see the focus of the microscope changes. So these beads, this one here has a single tether and comes up very high. This one here has a double tether, doesn't come up as high. And this one here is stuck completely to the surface. So this is just demonstrating that here we have one good bead, as it were, to work with. Well, with Martin Stops in the prototype, sort of building electronics, he made a system which actually has two magnets. So we now have a polarized magnetic field, so we can rotate the beads. And so by rotating these two magnets around, we can rotate the bead, and that enables us to twist up the DNA. Now that's important because remember, before the initiation process, it was all negatively supercoiled in the bacteria. So we want it to be in its sort of native state, all that sort of supercoiling in there. And so Algae has been using this method to deliberately supercoil the DNA. Now, this is where we have our lecture demonstration. I'm going to start off, and then my two worthy assistants are going to come up to complete the whole show. So this is now, this is a piece of DNA. So built large. So DNA, as you know, is a helical molecule, double helix, and we have it tethered at both ends. And what we can do is turn it. So I'm going to turn it now, and I'm going to put on what's called positive supercoil. So imagine turning that bead. I'm fixing it at this end, and I'm turning it at this end. Imagine now we're turning it round and round, and you can see now as I turn it, can you see I've reduced the pitch. It's become tighter. And actually, if you put on positive supercoil, you tighten the pitch of the Watson-Crick helix. And DNA doesn't like that very much. So what tends to happen is the whole thing then goes into this state, where it forms what's called a plectinine. And the more you twist it up, the more it coils. And that's that. Remember then that AFM image I showed you of that rather nice work by Claudia? That was actually what we looked at on the AFM, this supercoiled DNA. In fact, this is positively supercoiled. In life, it's negatively supercoiled. Now, negative supercoiling is where it gets interesting. And actually, this is where my two assistants will come to help. And I think, actually, since it's Algae's project, we're going to break with tradition, and I'm going to stop talking, and he's going to tell you a little bit about this ORI-D and the negative supercoiling business. So Andy is, he's a cover slip, and I'm a magnetic bead.
So now if we pull on DNA and slightly harder, and if we introduce positive DNA supercoiling,
okay.
So what we're doing now, we're actually increasing the pitch of the DNA. So in a way, we're actually tightening up the DNA duplex. But if we do the same to the other direction, so this time we supercoil DNA negatively, and now we reduce the pitch of the DNA, and it facilitates some local denaturation bubbles to form, while we have two DNA strands separating from each other. That means base pairing is disrupted for a moment. And when it happens, the DNA has two ways to go. It can either come back and form the DNA duplex, embedded complementary repeat in our DNA, what we have in ORID, then the DNA can form secondary structures, namely DNA cruciforms. And these DNA cruciforms are stabilized by negative DNA twist. They are stable at these conditions. And if we imagine, if we imagine the RevD protein coming to the ORID sequence and specifically recognizing it, and then we believe that these DNA cruciforms are somehow important for the RevD function. And what the RevD protein does, it probably stabilizes these structures even more, and then it cuts a single DNA strand, and that exposes a single-stranded region here. So the DNA is just about to be free to rotate around a single covalent bond in the phosphate sugar backbone. So when these two ends are released by the RevD protein, so basically the DNA supercoiling is lost.
Thank you. Well done. Very good. Right, your fee will be paid later. So that